everybody. Today we're going to take a much closer look at DNA, the molecule of life, deoxyribonucleic acid. This molecule is the key to understanding the flow of information in living things. Let's go ahead and get busy. So early in the 20th century, scientists were trying to find the molecule of inheritance. There was a scientist named Thomas Hunt Morgan who was working with Drosophila melanogaster, or commonly known as the fruit fly. And he was able to show that genes are located on chromosomes, those colored bodies within the nuclei of cells. And two major molecules wind up being in the race for being the genetic molecule. It was DNA and protein. Within the nucleus, these two are very closely intertwined. In fact, now we know that DNA wraps around proteins called histones, but that's for another lesson later. But as of right now, the front runners for the genetic material are DNA and protein at this time, early in the 1900s. Now, when you're working with something you don't know a lot about, you have to be very choosy with the organisms that you are working with. And part of the challenge of these experiments was determining the proper organism for this. So what he does, what a lot of scientists do is they wind up studying bacteria because they're very simple, they reproduce often, quickly, and there's a lot of them. And they actually wind up eventually looking at viruses that infect them to kind of seal the deal on DNA. But before we get there, let's go ahead and take a look at some previous experiments. One of the first experiments where we were trying to find the role of DNA was done by Frederick Griffith in 1928. He works with two strains of a pneumonia bacterium, one which was pathogenic or disease-causing, and the other one which is just harmless. They're two different strains of this pneumonia bacteria. What he winds up doing, bottom line in his experiment, is he mixes heat-killed remains of the pathogenic strain. So he winds up killing the bacteria with heat, the one that causes disease, but then he mixes it with normal bacteria, and what happens is that the normal bacteria become pathogenic. And he calls this phenomenon transformation, which is defined as a change in genotype and phenotype due to the assimilation or the uptake of foreign DNA. Let's take a look at his experiment closer. So you can see here, he goes ahead and he has living S cells, these right here, and these have a smooth appearance under the microscope, and that's due to the capsule that surrounds them. Now, this is a pathogenic bacteria. He injects it into a mouse, and the mouse winds up dying. There's also the other strain, the harmless strain, which we can call the control, and that strain here doesn't have a smooth capsule. Instead, it looks actually kind of rough on the outside under the microscope. He injects this bacteria into the mice, and the mice live. Then he tries it another way. He kills those S cells with heat and injects what's left over into a mouse, and the mouse stays healthy. And then his last attempt here, he goes ahead, he uses those heat-killed S strain cells, but then he mixes them with the rough strain cells, which are harmless, and he finds out that they wind up becoming transformed and killing the mice that he injects them into. So what does this mean overall? Well, something had to happen to let the genetic content out of a heat-killed cell and get into the rough strain cell and take on its characteristics. So what essentially occurred here, we saw that the bacteria was ruptured by the heat, and what we have now is fragments of DNA that are leaving the cell, and nearby cells that may be a different type, such as the rough strain, are taking in those fragments of DNA and incorporating them in part of their own DNA genome. Now, in the case of these two cells, that means that the pathogenicity trait, the ability to harm organisms that it infects, is now part of the rough strain. It has been transformed. Another experiment was done by Avery, McCarty, and McLeod, and what they wanted to do here, because Griffith's experiment did gain some traction, it still wasn't readily accepted as far as which molecule is actually passing on the traits to the next set of organisms. So in 1944, an experiment takes that even further. So if you take a look at our picture here, what we have is we have heat-killed virulent strain bacteria, which wind up in the in this Erlenmeyer flask here, we'll call it S strain, the smooth strain, which is a virulent filtrate, which means if that gets injected in something, it's going to hurt that thing, okay? It's going to kill anything that it becomes injected into. Now, they're going to go ahead and they're going to treat this sample of S strain virulent filtrate with a number of enzymes, and you can see them here. There's one called RNase, one called protease, one called lipase, and one called carboase. Now, how do they get these names? Well, the ASE ending on them means that they go to work on those specific types of biomolecules. Now, notice that I kind of left out DNAs in the middle, and I did that for a reason. So let's see what happens. He hits that S strain filtrate with the RNase, which destroys all RNA. He then adds the treated samples to cultures of R strain, or the harmless bacteria, and what he finds out is that it changes it to both S and R strains. So this is pathogenic. He then, this group of scientists then go and they treat their filtrate with protease, which destroys the protein in that sample. And once again, treats the samples with the R strain, and we wind up with both S and R once again. Lipase destroys lipids, and if we treat the sample with, in this case, with the R strain, we then get the S and R again, another pathogenic sample. So far we have 3 pathogenic samples. And the carboase destroys the carbohydrates, and we treat it again, and again we get the S and R strains. Now, with the DNAs, with the ASE ending, that means that enzyme destroys DNA. So when you add that treated sample to the culture of R strain bacteria, we don't find that S strain exists anymore. All that's left over is the original R strain that is harmless. So what that means is by destroying the DNA, you took away its ability to change this one here into a pathogenic strain, which again added weight to the fact that it's DNA and none of these other, especially protein, that are the genetic inherited material. Now, believe it or not, even with this experiment, it was still hard to accept that something made of 4 letters, something so simple in molecular shape and structure, could be the genetic material. After all, proteins are made of 20 different amino acids, which is, there's a lot more ways to arrange 20 amino acids than there is 4 nitrogenous bases. So it was still pretty hard for scientists to accept. Their conclusion was based on experimental evidence that DNA worked in transforming the harmless bacteria into pathogenic bacteria. That's kind of like the bottom line here, okay? So let's go ahead and see what really seals the deal as DNA and nothing else. When you pit DNA versus protein in a separate experiment, this is going to happen. We mentioned earlier that viruses and bacteria were used in order to further the knowledge that DNA was truly the genetic material. Now, viruses, we haven't quite got there yet in our class, but we can take a look at some of the parts and learn the anatomy of a very simple virus right now. So let's go ahead and add some parts to these viruses that we see infecting a cell. First, we have what's called a phage head. This particular type of virus is called a bacteriophage, or just phage for short. We have a tail sheath, which kind of covers up the internal machinery of the virus. We have tail fibers, which are kind of like the landing platforms onto the surface of a cell. And then we have DNA that gets injected from the phage head down the core of the center of the DNA and into its host cell. I mentioned a second ago, those viruses are called bacteriophages, which we use often in molecular genetics research. In 1952, 2 scientists, Hershey and Chase, are going to perform an experiment that pits protein against DNA, which is finally going to get us that understood information that we are looking for, that DNA truly is our genetic material. And their experiment kind of went something like this. What you see in brown in our picture, this is a bacterial cell. We'll kind of label it as we go on. You see one below as well. And you see a couple of phages on the top that are currently in the infection producing stage, where they've landed on the bacterial cells and they're injecting their DNA into that bacterial cell. DNA resides in the head of the phage, and you can see that in this one here, it's already began traveling into the cell. And this one here is also made of a radioactive protein coat. And what you can see here from this flask is this is going to be some radioactively labeled protein. And I'll go ahead and you'll see we often call it hot protein. And this one here has a radioactive isotope of sulfur 35. And what we know about protein is that there are disulfide bridges in protein. So everywhere where there's going to be a disulfide bridge, you're going to have this hot radioactively labeled sulfur as part of your protein, which is then going to make up the coat of your viruses if you grow them in there. Now, the other part of the experiment is using radioactive DNA in the phage head. If we go ahead and we use a radioactive isotope of phosphorus 32 and we grow viruses in a sample, then only the DNA is going to wind up being hot in this case. The protein will remain cold protein that doesn't have anything radioactively in it because you don't have phosphorus in protein. Instead, we have it in our structure of DNA. So that truly identifies a DNA sample versus a protein sample. Sulfur is readily present in all protein, and phosphorus is readily present in DNA. So what's going to happen is he's going to take samples of these bacteria from each case, one with the hot protein, one with the hot DNA, and he's going to run them through a blender. And after running them through the blender, we're then going to analyze what comes out after we centrifuge our samples. So after centrifugation of the samples, what we find is that the heavy stuff drops to the bottom in a centrifuge tube and the lighter stuff winds up on top. So the pellet is going to contain bacterial cell and its contents like DNA. And what's going to wind up above are chopped up pieces of the phage protein, which we can clearly see are, in this case, they're the hot type. That means they're radioactive. In our other sample, we're going to do the same thing. We're going to centrifuge our sample. Again, the protein ghost pieces are on the top, and our pellet down here, which is hot, we're going to go ahead and we're going to analyze it, finding that only the DNA, which wound up in the pellet, winds up being our radioactive sample that we're looking for. So in this case here, the contents of what was inside those cells winds up in the pellet, and that is going to be our hot DNA, which is going to prove without a doubt that it's DNA and not protein that winds up being passed on to the next generation. Here's another view of that sample. Again, we have sulfur growing the viruses in this case here, and in this case we have the hot phosphorus, the phosphorus 32 isotope. In both cases, samples are blended. In this one here, we find out that our liquid on top of our pellet, what didn't get into the cell is our hot protein, and what got into the cell, what winds up in our pellet down here is our hot phosphorus sample, again showing that DNA is truly our inherited molecule of genetics. Around the same time in 1950, there were more experiments done with compositional data of DNA from one species to the next. Erwin Chargaff, a scientist, he did some experiments where he analyzed the composition of DNA, and what he was able to show is that the composition changed from one species to the next. That is, not all species had the same amount of A's and T's versus G's and C's. But what he did show was that A and T was always equal to G and C. So those are going to be named Chargaff's rules, saying that in every species, regardless of complexity, we see that A is equal to T and G is equal to C. So that evidence of diversity made DNA a more credible candidate for the genetic material as well. And with the Hershey-Chase experiment, it pretty much nails it down. Here is an example of different species that exist and the percent of composition of certain bases that they have. For example, in human beings, we have around 31% of A's and T's and about 19% or so, mid-18s, of G's and C's. Drosophila melanogaster, you can see that a few ticks down in A and T content, but a few ticks up in G and C content. So there's a number of different species that you can do with this, of course, and every time you should probably wind up with a different amount. Up on the top here, we see some diagrams of our various nucleotides, where all three things are connected: our phosphate group, our sugar, and our nitrogenous base. The purine molecules, you can note, are adenine and guanine, and purines are characterized by having a double ring structure like you see here. Pyrimidine molecules are your T's and your C's, and they are characterized by having a single ring nitrogenous base. Okay. Oh, guanine, and I probably should have went this way with my arrow. There we go. Okay, so those three parts make up the nucleotides. We have two major classes of nucleotides: pyrimidines and purines. Purines have 2 rings, pyrimidines have 1 ring. During this time in Great Britain, there were 2 scientists, Maurice Wilkins and Rosalind Franklin, and they were using a new type of experimental procedure called X-ray crystallography to study molecular structure. And Rosalind Franklin is going to produce a picture of DNA using that technique, which you can see here. And what we're seeing is not something that looks like a strand of DNA. What we're doing is we're looking down the core of DNA, and we have these unique light and dark bands that we see. Now, it wasn't exactly known what we were looking at in terms of the DNA until a couple of other scientists took that photograph and interpreted it. So Rosalind Franklin provided the evidence that these other 2 scientists are going to use to nail down their theory of the shape of DNA. So in 1953, 2 very famous scientists named Watson and Crick are going to introduce their double helical model for the structure of DNA. And the hereditary information is going to then be known to be encoded in that DNA and reproduced by all cells of the body. DNA as a molecule will direct development of biochemical, anatomical, physiological, and to some extent behavioral traits. Think of animals that don't need to be taught anything, and upon being born, they know what to do. The only reasonable explanation for that is that it's encoded in their DNA. Their genes, which are stretches of DNA, code for certain proteins that will help them, enable them to survive. Franklin concluded that there were 2 anti-parallel sugar phosphate backbones with a nitrogenous bases paired in the molecule's interior. So that's what you see here. You can kind of see our phosphate backbone, and you'll notice that there's kind of a zigzag pattern here. That's a sugar phosphate backbone, and the other side has one too, but the other side it's upside down. And that's what we call the anti-parallel nature of the sugar phosphate backbone. We can take a look at it closer up in terms of how it bonds. We can take a look at space-filling models where chemists go to work on it using software that digitizes what those atoms actually look like. And here we can also notice that down the center core, you have a number of connections. These are hydrogen bonds. And you notice the AT connection has 2, and the GC connection has 3. So the GC connection is a little bit stronger than the AT connection. Now, hydrogen bonds are fairly easy to break, except when you have a long line of them. I like to think of it as a zipper. If I have one tooth on a zipper that's zipped and the rest is open, I can easily tear it apart. But if I zip the entire zipper, it's very difficult to pull that zipper apart. That's how I like to think of the hydrogen bonds. And those regions that have G to C connections are harder to take apart than those that have the AT connection. When they first were experimenting with designing this double helical structure, they took a look at a couple possibilities. One, that a purine and a purine would bond together. And what we find, and you can see it here, is that you're kind of exceeding the width of the DNA, which would make for a very lumpy outside. And we don't see that. So purines and purines wind up being too wide. If they put 2 pyrimidines together, what we find is it's too narrow. There's all this open space that you can see here and here. But if you wind up using a purine and a pyrimidine, those 2 together, you're going to wind up fitting the width of the DNA at a consistent width the entire stretch down. So purines and pyrimidines, the bonding between those 2 was just right. So Watson and Crick reasoned that the pairing was more specific, and it's based on base structures. And they call this the law of complementary base pairing. And what they determine is that A can only bond with T and G can only bond with C, and not the other case with the purines and the pyrimidines. And they go ahead and they actually use tin and cardboard cutouts, and they're able to go ahead and link the bonds between, in this case here, you have a nitrogen on this molecule and you have a nitrogen on this molecule, and you're able to form those hydrogen bonds as well between this nitrogen and this oxygen. They cut out the pieces, and they found that there was a hydrogen bond between this H and an O, between this H and the N, and this H and an O when you look at G and C, guanine and cytosine bonding together. So Watson and Crick model also helps reinforce Chargaff's rules, which A should equal T and G should equal C. So I hope you understand the molecule of DNA a little bit better now, both in structure and in terms of why it is the genetic material and not anything else like proteins, RNA, carbohydrates, or lipids. Hopefully those experiments also gave you some insight as the type of thinking that needed to be done in experimentation in order to show that DNA was that genetic material. Keep studying, and I'll see you next time.